So I'm only going to talk a bit about, well, certain aspects of image analysis, specifically for multiplexed imaging data, which is still relatively new, I would say. So directly up front, what I'm going to present here is joint work with Jonas Windhager. He's also on this call, so if there are Python questions, he might jump in. Yeah, he's a PhD student in our lab and yeah, excellent software engineer. So he really makes the life of a lot of biologists easier. So I will start with a quick introduction to multiplexed imaging. The concepts that we're using, they're coming from classic visual imaging. You are maybe familiar with immunofluorescent imaging and immunohistochemistry, where you have either fluorescently labeled primary or secondary antibodies, or antibodies labeled with an enzyme that creates a visual readout. The problem here is that due to spectral overlap, you can only measure up to 4 fluorophores at once. And there have been several groups working on kind of tackling this issue by different technologies. So there are two large directions you can go. One is kind of cyclic multiplexing, and another one is where you do use different tags to these antibodies. Here, cyclic multiplexing, you either have oligonucleotide tagged antibodies to which fluorescently labeled probes bind sequentially, or for I and CySIF, they use fluorescently labeled antibodies, but then they have this strip and stain protocol to multiplex up to, I think, 25 to 40 markers at once. The nice thing about this technology is that you can image a whole slide, but it does take some time for this cyclic strip and stain approach. The second approach is multiplexing via metal-tagged antibodies. So I'm a postdoc in the Bodenmiller lab. The lab developed imaging mass cytometry, which is one of the two techniques. The other one is multiplexed ion beam imaging. They are comparable in a way that they use metal-tagged antibodies and then either laser ablation or ion beam ablation and a type of light detector to detect heavy metal ions. This technology is limited by the field of view that it measures. It's relatively small, but we can measure hundreds to thousands of samples in a couple of weeks. So the one thing that all these technologies have in common is kind of the data type. So these technologies produce multi-channel images where these are grayscaled images, and every channel stores the abundance of a certain protein, or even by now we are detecting RNA in tissues. One of the first image processing steps also includes segmentation. So we want to find objects in these images. Most commonly, these objects are individual cells. And then by combining multi-channel images and segmentation masks, we export single cell data. So this is, for example, the mean intensity per marker and per cell, but also morphological features or interaction graphs. And so what I want to talk to you about today is a framework that Jonas and I developed over the last year or so that really goes from the raw data down to spatial data analysis. The first part will be mainly based on Python for image processing and visualization. The second part will be in R, also for image visualization and spatial data analysis. So Jonas has developed steinbock, which is a Dockerized framework for multiplexed image processing. The main input here are multi-channel images. Depending on the technology that you're using, these can be in a proprietary file format. So we are working with .mcd files. These can be also just TIFF files, which usually any reader function and then writer function can produce from your file type. Napari is quite powerful in visualizing these multi-channel images, but also single-channel images. And Jonas has developed a plugin that specifically works with our data. And I will show you this in a bit. Steinbock is a framework that performs image pre-processing. This is file type conversion, cell segmentation, quantification, and then data export. So this is written in Python, available as a Docker container, and produces single cell data. I'm more focused on developing Bioconductor packages to handle imaging, but also single cell and spatial data. I've developed cytomapper that reads in multi-channel images and segmentation masks for visualization, and I've developed imcRtools to read in single cell and interaction data to perform spatial visualization and spatial data analysis. We do also export, or Steinbock exports data in the ndata format, so this can be directly read into ScanPy or SquidPy. So I mentioned before, Napari is now quite commonly used to visualize multi-channel images. One of the issues we have with our data, it comes in a proprietary data format, but Jonas built Read IMC, which supports Napari IMC, a plugin to directly visualizing multiple aspects of our data. And I can show you this here. So this is a slide overview. And then we have these bright field panoramas of parts of the tissue. And then in these overview images, we select regions of interest that we want to ablate and measure. And here you can select an individual channel. This is by Menton in green, and visualize this on 2 selected regions of interest. Jonas here is selecting now a different channel. I think this is SMA, and overlays it on the measured regions of interest. Of course, multiple channels. So I have to say we measure 40 proteins at once, and multiple channels can be visualized here at once. It is quite reactive, and all the settings are shared across regions of interest. And then, of course, users usually measure multiple regions of interest and multiple panoramas. All of these can be interactively visualized. So a couple more details on Steinbock. So as I mentioned before, imaging mass cytometry doesn't come in the standard TIFF format. One of the first steps is to pre-process it into TIFF format. Steinbock also works with a so-called panel file in which you can specify the channel names of the, in our case, metals or fluorophores that you use for measuring. You can also specify there if you want to use these channels for image segmentation. Once you've generated TIFF files, there are 2 ways of segmenting these images. With segmentation here, I specifically mean single cell segmentation, but of course there are also other segmentation or other objects you can detect in images. One route is using DeepCell. This is a pre-trained neural network that basically works out of the box within the Steinbock container. Another more classic route that we used in the past is to label pixels in images based on if they are part of a nucleus, a cytoplasm, or the background. We use Elastic to train a random forest to then perform pixel classification. And based on the pixel probabilities, we use Cell Profiler to then segment individual cells and images. No matter which approach you use, you get segmentation masks out at the end. These are single channel images where sets of pixels with the same ID represent individual objects. Steinbock then takes these masks and the multi-channel images and measures intensities. So this can be the mean pixel intensity per cell and per channel. It also measures shape features of the cells and if cells are in close physical proximity, and stores these measurements as CSV files. Data export here means that the measurements can also be exported in, for example, the N data format. It is available as Python package. For new users, we usually recommend using it via the command line interface by using Docker in the background. And here I've just listed a couple of commands. These are all commands you need to really come from raw data down to single cell features. So you set an alias, which basically specifies the Docker run command. Pre-processing is only needed if you don't have TIFF files available. If you have TIFF files available, you can directly segment these images using DeepCell. And then there are 3 measurement calls that measures the intensities, the shape features, and cells in close physical proximity. It then produces a number of output files. It is quite standardized. That's why I was able to write a reader function, which puts all of this data together in a spatial experiment object. This then brings me to the R or bioconductor side of things. So after image preprocessing, you have multi-channel images in TIFF format, segmentation mask in TIFF format, and then a number of single cell measurements like intensities, shape features, and interaction graphs. I developed cytomapper to handle multiple multi-channel images and segmentation masks in R. This is used for image visualization and cell visualization on composite images. And I've developed imcRtools to read in Steinbock exported data into a spatial experiment or single cell experiment for then spatial visualization and spatial data analysis. We also have a more extensive R-based workflow available online that summarizes and gives guidelines to all these analysis steps. I will first talk about cytomapper for image visualization directly in R. So it allows the user to visualize cell-specific metadata on segmentation masks. So you can see here these small objects. These are individual cells that we segmented, and they are now colored by cell type, for example. You can also visualize mean cell intensity or other features, other expression features that you want to measure on segmentation masks. You can also use it to directly visualize pixel information. So here you can see a pseudo color composite image of 3 of the measured markers. You can outline cells on images. I will mention later again, this is quite nice to check segmentation quality control. And we also provide a Shiny app that allows you to gate cells and then visualize gated cells on images. I will talk a bit about the architecture of cytomapper. So it uses 2 data classes. One is the single cell experiment. It does also work with the spatial experiment, but it wasn't available back at the time. The other object is called the cyto image list data class. I had to write this to efficiently handle multiple multi-channel images where channel information is shared across all images. And it mainly exports 2 functions. The first is plot cells, which visualizes segmentation masks. Here, the mask object is a cyto image list object that stores multiple segmentation masks. And object here is a single cell experiment that stores all available expression or expression information or metadata related to the cells also found in the segmentation masks. To link these two objects, the user will need to specify a slot called image ID, which can be found in the element data of the cytoimage list object. It's just a simple list from the S4 vectors package. And the same entry can also be found in the call data entry of the single cell experiment. This is how cells are matched to images. Cell ID needs to be specified so that basically a cell ID, which is usually an integer, is linked from the call data of the single cell experiment to a set of pixels with the same ID in the segmentation mask. And here, for example, images are colored by cell type. The second function is plot pixels, which is a lot easier. Here, the main object is a cytoimage list object, which stores multiple multi-channel images. And then by specifying up to 6 markers, you can generate these pseudo-color composite images of marker expression. During the revision process of the paper, one very critical point came up, the question about scalability, because images can be quite big, and keeping all these images in memory is just not feasible. And here's an example. On the right, you can see a parallel visualization of 100 of our images, and they are even quite small. So every image is 500 by 500 pixels. The segmentation mask, which are just single channel images, are 200 megabyte in memory. Multi-channel images, in this case, these are 38 channels, are nearly 8 gigabyte in memory. So as an extension to cytomapper, I switched from using the image object from EBImage to an HDF5 array object from the HDF5 array package, which basically links images that are stored in the HDF5 file format on disk. Cytomapper provides the load images function. The default works in a way that it reads in TIFF files, for example, and then if you want to store these images on disk, you can write them out as HDF5 files. By now, it can also read in data sets directly from HDF5 files, so you will never need to read in this imaging data into memory. So even hundreds of images can be linked within seconds, and you're ready to visualize them in R. So now a couple of use cases of cytomapper. So one which people in the lab also quite regularly using is for segmentation quality control. So after image segmentation, you want to make sure that you actually detect individual cells and images. Segmentation is kind of a unsolved challenge in a way. There's never a perfect segmentation. So the most straightforward way of checking is to just visualizing the outlines of cells on composite images. And you can do this by using the plot pixels function, which produces, in this case, a 3-color composite image. If you also provide a segmentation mask object, it will just outline individual cells based on the segmentation mask information. A nice thing about working here with the single cell experiment object is that it's subsettable. So you can specifically visualize cell types on images. Here's an example. This is a single cell experiment object that contains all cells of my experiment, but I only want to visualize beta, alpha, delta cells. These are, this is pancreas, and 2 sets of T cells. So I subset the single cell experiment object, and I call the plot cells function to visualize cell types on the images, and it only visualizes the selected cells. All other remaining cells are colored in white, which is specified by this missing color parameter. In a deference setting, and this is usually used if you want to figure out if you actually phenotype the right cells. You can subset the single cell experiment to only contain one type of cell, and then outline cells based on their segmentation mask. So here on the right, you can see I'm only outlining cells that we detected to be the T cells. And this looks quite nice. Well, this is actually data from Nicolas Damon from our lab, and there was only one cell missed here. This is really kind of the powerful feature of multiplexed imaging data, that you can look at the data and see if you've characterized the cells correctly. The last function of the cytomapper package I want to show you is a Shiny application that we wrote to gate cells based on the expression and then visualize the cells on images. This all came from one of the big problems we have, that clustering does not always work to really find clean cell phenotypes. So for single cell RNA-seq or cytoph data, it's usually a bit more straightforward to find the major cell types. Due to lateral spillover and missegmentation in our images, certain cells always get kind of marker intensity from neighboring cells. So we use a probabilistic classification approach instead of clustering. But for classification, we need some sort of ground truth data, which can be generated using this Shiny application. So you can directly load it from R. It opens a Shiny app. You can select a number of gates that you want to set, and the images that you want to gate on. If you want to gate on the raw count data or some sort of log transformed data. And then here I'm gating on equaterin and CD3 to find T cells. And then again on CD8 to find cytotoxic T cells. In the second tab, you can then visualize the images, select the markers, adjust the contrast, zoom into these images to see if you selected the right cells, and then save it again as single cell experiment object while storing the gated cell label. And for the design choice to do this per image, we usually observe small staining differences between images. So if you would start gating across all images, you introduce biases in that way. And in that way, you generate some sort of ground truth data set for classification. Yeah, then coming to the second bioconductor package that I wrote to kind of support the analysis of highly multiplexed imaging data, but also in this case, any sort of spatial data. This is called imcRtools. The read steinbock function directly reads in the standardized output from steinbock, and it produces a spatial experiment object or single cell experiment object. So steinbock does already generate spatial object graphs. So these are basically edge lists where every edge indicates two cells in close physical proximity. But if you want to create these interaction graphs on the fly, imcRtools provides the build spatial graph function, which takes a spatial or single cell experiment object and then builds a spatial KNN graph or via expansion or via Delaunay triangulation. And this graph is stored in the call pair entry to the object. For visualization, imcRtools provides this plot spatial function, which visualizes the centroids of the cells. And you can set any sort of features of the cells. You can also draw these edges between cells. This is quite useful if you want to observe kind of the range of the spatial interaction graph that you've constructed. You can also color the edges based on metadata of the from node. The first spatial data analysis function that I want to present here was actually, or the idea was developed by Gary Nolan's lab. It is relatively simple. You start with this interaction graph, and then for each cell, you aggregate the direct neighborhood. So you can either compute the proportion of cell phenotypes in the direct neighborhood, or you can summarize the expression counts in the neighborhood. And in imcRtools, this is done by calling the aggregate neighbors function. And then in the aggregate neighbors slot of the call data, you can find this nested data frame where every column, in this case, indicates the proportion of a certain cell type in the direct neighborhood. And in this second step, you can then use these features to cluster individual cells. Usually we use k-means. We need to look into detail again if there are also graph-based clustering approaches that we can use to cluster cells. But what you usually get out here on the right side are cells that are similar based on their direct neighborhood. So again, I'm presenting pancreas data here, and then all these orange and red cells are part of the pancreatic islets. The blue cells are clustered immune cells, and the green cells are clustered exocrine tissue. Here, this blue patch is quite interesting because it contains most of the T and the B cells. Yeah, so this cellular neighborhood detection function is relatively unbiased. imcRtools also provides the patch detection function, which was developed by a former master student in the lab. It's supervised in a way where the user needs to provide some sort of cell phenotype, and the function will detect connected sets of cells with this exact phenotype. After patch detection, the function will then construct a concave or convex hull around the patch, and this can be expanded to directly also incorporate neighboring cells. Here, this is one example of the patch detection function where I specify all cells of pancreatic islets, and the result looks like this. So on the left side of this plot, you can see pancreatic islet patches. So all of these cells are part of the islet plus I think 10 micrometers surrounding the islet. On the right side, I detected immune cells that are in close proximity. So let's say fully connected sets of immune cells. And then finally, the last function that I want to present here is the test interactions function. This was developed by Dennis Shapiro in the HistoCat paper. The idea here is to detect cell types that interact more or less frequently compared to what you would expect by chance. And this is done again by constructing an interaction graph or using the one exported by Steinbock. And you first count the average number of cell type interactions per image. And to get an estimate if these average interaction counts are significant compared to a random distribution of cell types, the function will create an empirical null distribution by randomly randomly perturbing the cell labels of the image and therefore deriving empirical p-values. This is something very similar to also what ScriptPile is doing, I noticed. The output here can be visualized in form of heat maps, where the rows and the columns just indicate the cell types. The fill entry here is the average interaction count between these cells, and the little stars indicate significance compared to a certain threshold. Green means the cell types interact more often compared to what you would expect by chance, and the red stars indicate cell types that interact less frequently. We never really look at individual p-values per image, but we usually aggregate them across multiple images to figure out if certain cell types interact more frequently in a certain condition compared to another condition or tissue type, for example. Yeah, so I'm already getting close to the end here. This is just an overview on the next steps that I had in mind for cytomapper and imcRtools. So I'm still not quite happy with the way of visualizing images in cytomapper. It's still relatively slow when you need to draw all pixels in the graphics device. So I'm thinking about some sort of browser-based interactive visualization where maybe low resolution PNGs are written out to disk in the background. It would also be nice to kind of merge visualization of segmentation masks with spatial object graphs, still in one of the issues, but I didn't get around to work on this yet. For imcRtools, it would be nice to implement some concepts of spatial point processes. So the K and the L function are quite regularly used when looking at co-occurrences of cell types or some sort of clustered cells. There the question is also a bit if we need new data classes to efficiently handle these sort of multi-scale functions and their visualization. Also a bit related to this are kind of the open challenges that I'm seeing, specifically image analysis in R. So cytomapper can read in TIFF, PNG, and JPEG directly into memory. I know that raster is pretty good at using an on-disk version, or basically supports on-disk handling of TIFF files. But from what I've seen, this is only single channel so far. That's why I used HDF5 as file format to directly use the HDF5 array package as an on-disk representation of multi-channel images. But again, these files need to be written first. So I'm discussing with Jonas that steinbock should also support writing of HDF5 files, which might come in the next release. And then, of course, there are more efficient file formats for imaging data, like SAR and N5. And I think some guys from EMBL also presented this OME NGFF file format for which data handling is not really available in R at the moment. As for image visualization, I like the SVG Lite and SVG Pan Zoom package for interactive visualization. However, that's a bit tricky to really control the resolution of the images. This is really the strength that Napari has, that as soon as you zoom in, you keep a certain resolution of the images by using these multi-scale image formats and ask image. Yeah, with this, I want to thank the whole Bodenmiller lab. Of course, Jonas, who developed steinbock and Napari IMC, and of course, a lot of other tools in the lab, Bernd for the supervision. Vito was a PhD student in the lab who worked a lot on the basis of all the data analysis approaches. Nicolas developed the first version of cytomapper, which was really cool. Jana, Tobias, and Daniel provided code support for imcRtools. And thank you for your attention.
Thank you very much, Nils, for a fantastic overview and a fantastic talk. Really a lot of very nice functionality and infrastructure for spatial omics analysis. At this point, I'd like to invite everyone from the audience. If you have questions for Nils, just feel free to raise a hand, as people are already starting to do, and unmute yourself or ask in the chat. I see Artem has a question, if you just want to speak up.
Yeah, sure. Nils, very nice talk. I had a question about the technology. So some of the early images you showed had the darker borders around the individual tiles. So in immunofluorescence, this is usually due to uneven illumination. And I was just wondering what the kind of the physical process is for IMC data that gives you that. So that's part one. And then second part of the question is, can you still use the same, quote unquote illumination correction methods to correct for those dark borders? Mm-hmm. It's where you were showing Napari IMC, there's a kind of a darker pattern.
You mean in this video?
Yeah.
Yeah, so I know what you mean. We usually don't really have these effects, as we don't measure the whole region at once, but we do this pixel ablation. So a laser shoots basically into the tissue at a 1 micrometer resolution, and the material will then be ionized and led into this time of flight detector. So we actually never really have to deal with illumination correction. But you're right that for fluorescently,
So I mean, I am seeing these dark borders, right? Dark patterns. So what is causing that?
Here in this image, you mean?
Yep. Yeah, like on the gray channel, right? Do you see the dark grid?
So yeah. Okay. Yeah, now I get it. So the gray channel that you're seeing is a bright field overview of the image.
Okay.
And this is measured in, so this is, I'm not sure if you can see, do you see my mouse?
Yep.
Yeah, so this is just one image. So it will be stitched together.
Yep. So yeah, if it's bright field, then you okay.
But we never really work with these overview images. It's just for us to get an orientation where we are in the tissue.
All right, we have another question from Jacqueline.
Yes, Niels, thank you so much. I as an immunologist who spent countless hours counting cells by hand, I can't thank you enough for developing kind of technology. I wish it was available 12 years ago. So I have a couple questions. So as you mentioned, typically we like to take, multiple images, so like 20x or something. So just logistically, are you saying that we would take, maybe like a 5x image and submit that? Is that the input, or is the input 20 TIFF file, the 20 TIFF files that we took? And then I have another question after that.
Yeah, so here with cheddar, I really mean a different marker in a way, right? So for IF, this would be a 3 or 4 image, 4 channel image. Or is this not what you meant?
Yeah, so we typically like to count, if I'm counting cells, I'll take 20 images and count cells from 20 different places along the slide, so that I get a good representation. So the output then is I have these 20 TIFF images that I would then have to go and hand count the cells.
Yep.
So would I instead then submit those 20 images to you, or would it be submitting like an overview of the tissue, like at a lower magnification?
Okay, then I got it. No, so you would be submitting the individual images, but they will all be contained in this image folder here.
I see.
So for Steinbock, for example, I mean, this would work if you have 20 times an image where you have a Darby signal and some sort of membrane signal, for example, or multiple membrane signals. And then you can have as many images as you like in this image folder. And you can just call a single command, and Steinbock will go through every individual image and then perform some sort of the segmentation. Yeah.
Gotcha. And so that's great. So my second question is around, these images that you've shown are beautiful, beautifully stained, but knowing that in the lab that doesn't always happen, and often we have students doing it, and there's, whether it's bright field, background, they let it develop too much in the background tissue, there's background on the tissue, or if there's some autofluorescence from immunofluorescence. And so how are you like quantifying the intensity? Is it relative to the background tissue intensity, or is it a threshold of what you would expect based on the type of stain that you're doing?
Yeah, so in the images that we're producing, we don't have specifically this problem, but I'm also working with multiplexed RF data where you have all these, you have some background stain, how you really, so Steinbock works quite well, kind of ignoring all these issues. The segmentation still works quite nicely. The question at the end is just, if you look at these mean intensity values, can you interpret them across images, for example? And I know that other groups, they try to find some sort of threshold per image to then first identify positive and negative cells, and then only work with the phenotypes. And this is also what we are doing. If we observe very large sample effects, or batch effects, we try to first correct them in a way, but directly perform clustering or classification on some sort of low dimensional representation of the data, and not really on the counts or the intensities anymore. This is how we, so by correcting across all images first.
So similar to how when we're analyzing flow cytometry data and we set the gate, so you can say what's positive at the outset before you start analyzing the whole image.
This is what collaborators with multiplexed images are doing, right? Which is also really difficult because you need to look at every channel, at every image individually. So we worked a bit on algorithms that identify the perfect threshold. But at the end, we always had a user to correct it in a way.
Thank you.
Sure.
All right, thanks, Jacqueline. And we have another question from Friederike.
Yeah, hi, thank you. So yeah, I guess following a little bit up on what Jacqueline said, I was wondering, and you mentioned it, what types of QC do you typically do for the cell segmentation? So is it always just manual, hands-on gut feeling, oh yeah, this looks about right? Or are there some parameters and quality values that are being calculated that one can look at?
Yeah, so in this workflow that I'm currently writing, there's a whole section on kind of quality control, and of course segmentation quality control is one of the big things. It is really difficult though, because you don't know what the correct segmentation is. So after looking at the images, we also start visualizing the expression of cells as some sort of heat map, and then identifying features that overlap, which are not supposed to overlap. Basically just really also looking at scatter plots. If you only have 40 features, then you have an idea on how they should be expressed. In our case, we often see that CD3 and CD20, so T cell and B cell markers overlap, which are not supposed to be. But this is just because B cells and T cells are quite physically interacting in these tertiary lymphoid structures. And then we usually try to adapt the segmentation approaches to resolve this issue as best as possible.
Got it. And then just a side note, so what specifically is the difference between cytomapper and imcRtools? When would I use one over the other?
So cytomapper, if you want to work with images, and imcRtools if you only want to work with single cell data.
Got it. So imcRtools is basically just working with the matrix, kind of discarding the image part. Got it.
Right.
Okay, thank you.
Sure.
All right, the next question comes from Robert.
Yeah, great talk, Nils. I was wondering your thoughts on like how do we, what are good experiments to think about sort of collecting so that we have a reasonable resource for, you know, sort of applying machine learning type approaches to resolving some of the, you know, issues that you're challenged with, right? QC, cell type identification, et cetera. Have you, or the Bowden Miller lab thought about how to do that, and maybe comment on what data sets you guys have made public or are planning to make public?
I don't think I have it on one of these slides. So about a year ago or so, we started working on the IMC datasets package. It's an experiment hub package or bioconductor package that now contains 4 or 5 IMC datasets. But of course, it's only one technology. It would be nice to have codec, CySIF, 4i, any sort of multiplexed imaging technologies in there. So there are attempts. For example, the original paper that presented Deep Cell or the Mesmer model, they collected a lot of different data sets. I'm not sure how they make them available. The issue is just a bit kind of data storage and also really accessing this data.
I think question, so I totally agree that big data is going to be a challenge for us, and we have to find solutions for not shifting image data all over the place. But I think it's more, how do we get well annotated data sets, right? It's not hard to find an image. It's hard to find an image where there's some notion of ground truth. And most machine learning approaches require, sort of in some sense, the more data for which you know the truth, the better off you are in terms of trying to improve sort of how the process actually works.
For sure. So in our specific case, we provide these images in form of cytoimage lists, and then we provide a corresponding single cell experiment object. And then via this image ID entry, you can link any sort of metadata, for example, the clinical metadata of the patients from which the images were acquired, but also the proportion of cell types directly to the images. But this, of course, is only kind of image level annotation. It would also be nice to have some sort of pixel level annotation, which we've done in the past to, indicate if certain pixels come from the cytoplasm or the nucleus, or if there are some sort of larger structures available. This is what we don't provide at the moment.
All right, thanks. I also wanted to direct a little bit attention to a question in the chat, which I think has already been answered by Jonas, but I found it interesting as I'm also dealing with these C stacks in the context of Murphish data. So Alex asked, can Steinbock handle data sets with multiple C levels? And although that seems not to be possible right now, I found it quite nice to hear that this is something that might be possible in the future. On the other hand, Nils, maybe a question for me from my side. So we saw a lot of duality of single cell experiment and spatial experiment on your slides. So a bit more of a general question, do you see there any barriers on your end to broader adoption for spatial experiment? My understanding is that a lot of the functionality that you developed or started to develop at this point, spatial experiment was just not around yet, but it figures that adoption will be possible, for example, moving the cytoimage list over to the image data slot of a spatial experiment. So I just wondering what your thoughts are on that, whether you are having interactions with these spatial experiment developers, as you're having here a number of really nice use cases that these folks would certainly, I think, benefit from in their developments.
For sure. Yeah, it's a good point. So when I started writing imcRtools, there was the point when spatial experiment just came out, and I discussed with Dario a bit, actually via GitHub issues, how to kind of synchronize image handling. Back then, and I'm not quite up to date, I think the spatial experiment only really supports raster images, which I don't think supports kind of multi-channel visualization so that you basically adjust the levels of an individual channel. So you can visualize, let's say, H&Es or some sort of bright field images, but then visualizing multiple markers as composites is not really possible with this data structure.
And then maybe a last question from my side, if there are no other questions from the audience. I think that is a question that you most likely hear a lot. So if I understand you correctly, a lot of the functionality that is available in cytomapper and imcRtools is for analysis of spatial proteomics or spatial profiling of a handful of marker proteins. I wonder your thoughts on to which extent do these concepts also apply, of course, to the analysis of spatial transcriptomics. And I think especially in imcRtools, these nice concepts, these statistical concepts, these methodological concepts that you introduced, such as these neighborhood permutation testing and patch aggregation, they seem to kind of naturally apply also to spatial transcriptomics analysis.
Yes, for sure. Yeah, so you're right. In terms of images, I don't see there any application of cytomapper to spatial transcriptomics data, because also the way you would quantify the images in MERFISH or SEQFISH. The spatial data analysis functions that I've presented, they are directly applicable to spatial transcriptomics data, because we are only working with cell types, or let's say spot types. I mean, yeah, perturbation would also be applicable for Visium, but I think in fish type spatial transcriptomics data, it would actually be quite nice to apply these. You just need some sort of cell types at the end.
All right. If there are no other questions, then I would really like to thank you, Nils, again for a fantastic presentation, a very interesting discussion. Thanks for coming over, at least virtually via Zoom. And I just wanted to say that the spatial seminar series will continue next week with Fabian Theis on SquidPy, and in 2 weeks from now with Artem Sokolov on MC Micro. And with that, I'm wishing everybody a great day and hope to see you next week. Take care.
Thank you so much, and see you next week then.
Thank you, Nils.
Thank
Bye.
you.
Bye.